Hi, Miss Warren here, and I'm going to give you the directions for how to do the cell theory lab. All of the directions are written on this paper. They're taped at the instruction stations, they're at your tables, and they're at your lab station. So make sure, number one, that you read the directions. For part 2 of the lab, you're going to be looking at a sample of pond water. It'll be in a jar, kind of like this, only it'll be a lot dirtier looking, and that's actually good. All that dirt and dead and decaying material is what the little microorganisms live off of. Okay. Again, you're going to take a very clean slide. The cleaner the better. This one's actually kind of dirty, but get it really clean, dry it really good, polish it up with a paper towel so you have the best chance possible of spotting the microorganisms that are living in that drop of pond water. Okay, and the trick here is to get a sample. So this is nice clean water out of the tap. But when it's pond water, you'll notice kind of some schmutz on the bottom, some dirt or mud or plant material on the bottom, and then a lot of times the animals and cells are floating up at the top where there's a lot of oxygen. So those are two good places to get your sample from. So you're just going to squeeze in a little bit of liquid from that. If you can see a little bit of dirt or plant material in your dropper, that's a good thing, and because there's probably some things living on that. And you're going to take your microscope slide and you're going to put one good drop of pond water on there. You're going to put a cover slip on it and you're going to look at it. Now the trick is when you look at this, you're going to look at it under low power, maybe medium power. Get it focused, and then you're going to use your thumbs to move the slide around on the stage and look for movement. We're not using stain this time, so you have to look for living organisms that are actually moving around. A good trick is to look at the non-moving dirt or plant material that's in there and then look for the organisms around that. If you spend a good 5-10 minutes looking, you clearly have your microscope focused but you're not finding anything living, take your cover slip off, dry your slide off, and remake your slide with a sample, different sample, and see if you have any more luck. Use the time that you have. When you do find something moving, try to do a quick sketch of it in your notebook, and then also use one of these handy dandy keys that will be at your lab stations to help you try to identify what it is. Not everything is on here, but this will be a good first place to start. When you're done, you're going to take the cover slip off, throw that in the trash, wash and dry the slide, and put it back, just like we've done with our other slides. This one does not have to go in the bleach since it doesn't have human germs on it. Okay?